So you can kind of see the theme that's developed today for those of you that have stuck through the whole program. I mean, you know, what we're really trying to convey is the complete continuum from basic science to applied science to translational work. And what we're going to end on are two programs that are part of our translational program. And both of these were funded a couple years ago now in the large-scale competition that Genome Canada had in forestry and environment. And the first one is a project that's, the PI is Richard Hamlin, and the second project is, the PI is Judy Isaac Renton, and her co-PI is Dr. Patrick Ting. And so, and then for the panel discussion, what we're going to have is another individual that comes from a science background and a research background, but now is with the Ministry of Forests, and that's Harry Cope, is going to come and join us and talk a bit more. We're going to work some of the regulatory aspects of things into this. So let me just start out by making saying that Richard Hamlin is not going to be able to join us today. He called us, well, he didn't call us, he sent us a message yesterday morning, and this flu bug that's going around got him yesterday morning, and so unfortunately Richard's down for the count, and so we were quite concerned about how we're going to fill this gap, and so we sent him back a message, and even though I don't know if any of you have had this flu bug, but it's a nasty bug, it really just knocks you down, and we sent him back a note and asked him, is there somebody that could stand in for you and give your talk? And so what he did is he reached out in his, even though he was not feeling well, to Nicolas Feau, who is here today, who's a research associate with his lab and has been involved in the project for 1 year now. So Nicolas, we thank you very much for standing in for Richard, and welcome you to come up.
Thank you. I just want to add that so Richard asked me to do this talk today, and if you have any concern with the content or with my French accent, please email back to Richard. Not today. And not today. So I'm going to present you a general overview of our Taiga project that is funded by Genomics and Genome Canada since more than 1 year now. And Taiga is for tree aggressor identification using genomic approaches. And you can easily understand from this title that our objective is to help Canada to increase its capacity to identify exotic pests and pathogens from trees. This project brings together different institutions, multidisciplinary institutions, as academic institutions, as UBC and Simon Fraser University in Vancouver, but also some federal agencies involved in plant health research and plant health in general, and also groups involved in forest product innovations. Sorry, I have a gap. So, plant disease and more specifically tree disease are part of natural ecosystems. But there's two reasons why tree disease and broadly speaking plant disease can break into strong and damaging epidemics, which are, for the first one, tree domestication, with the intensive, dense, sometimes clonal plantation of tree, which represents some big targets for pathogens. And the second reason for plant disease and tree disease to break into epidemics is the introduction of alien invasive pathogens. And you can see on this graph the cumulative number of new detected alien species introduced in pathogen-free environments over 2 centuries. And the trend is clear, it's going up. And this trend can be directly related to the economic activities. The more economic activity you have, the more trade you have, and the more chance you have to a pathogen to be introduced in a new environment. There is on this slide 3 of the most infamous forest pathogens introduced in North America. They have 2 things in common. They are all non-native. They were all non-native in North America, and they cause ecosystem-wide. They strongly impacted ecosystems in which they were introduced. The most recently detected pathogen was the Sudan hog death, which reported for the first time 15 years ago in California, and which caused strong damage on oak ecosystem in this US state. Now, Sudan hog death is in Europe and has recently been jumped on conifers, and is specifically causing a strong damage on a large tree in England. Sudan hog death has been repeatedly detected in Canada, specifically in BC, and is still under strong monitoring all across Canada. So prevention and diagnosis in this context are key to prevent such introduction events. But the concern is that until now, disease diagnosis in plant pathology is mainly based on symptoms. And this methodology has been developed over 100 years ago and can only target non-pathogens. And you can easily understand that such a visual diagnostic or culture-based on petri dishes diagnostic of non-pathogens can have some failure in some case. Just let me definitively convincing you with this photo, this example of the Scleroderris canker on pine. We have 2 seedlings of pine infected with the pathogen Graminealia abietina, which is responsible of the Scleroderris canker. On the left one, we have a development of the symptoms because the seedling was placed under conducive conditions. By conducive condition, I mean the optimum temperature for the development of the disease. And the left one also carried the Scleroderris canker, but didn't develop symptoms, just because the seedling was not placed under conducive condition. There's no way, it's easy to understand from this photo that there's no way that visual inspection would pick up the seedling on the right. So, in this context of old-fashioned diagnostic, our project is to increase the Canada capacity in forest disease diagnostic and pathogen and specifically exotic pathogen detection and monitoring by making a link between the genomic data that we are developing and some application that we're going to provide to our end user. And these applications are diagnostic tool, validated diagnostic tool, and high throughput services for diagnostic that we will provide following a request from our end user. So our project is divided in 4 activities. The 2 first ones are related to the genomics, and the 2 last ones are related to application coming from the genomics part. So we're going to sequence, and we are sequencing some exotic and invasive pathogens of interest. It represents 3 groups of fungi and oomycete for a total of almost 20 pathogens. And the aim with this sequencing of these de novo assemblies is to run some comparative genomic job to identify core and unique gene sets to then plot them on bioassay on chips for doing diagnostic. The second time we also have a population genomics approach for 3 of the most important species which are sequenced in the first activity, and it represents almost for the 3 species 400 genomes resequenced, and the aim of this resequencing is to do some population genomics analysis and inferences of the route of migration and dispersion and introduction of these pathogens. So the 2 last activities are related to the diagnostic and the application of genomics to diagnostic. And so with the core gene sets identified in the first activity and the unique gene set, we're going to develop some diagnostic tool for our end user. And finally, the 4th activity is related to a gels activity, and which aim to inform us about the different potential issue and opportunity arising for the 3 previous activities. Just don't ask me questions about that, because I'm mainly a bioinformatist and genomics guy, so I will not be able to answer. Sorry about that. Okay, just let me know, showing one case study of what we are doing with the first and the 3rd activity of our project, and this case study is related to poplar disease. Everybody knows that poplar is an important resource in forestry, and specifically in North America, where poplar is widespread from coast to coast and south to north. It's also an important species in our landscape, specifically in riparian ecosystems. And from the economical point of view, poplar is sometimes in forestry compared to the crop species of forestry, with short rotation and production of important, highly valuable products as fiber, paper, wood products for construction, biomass, and even biofuel. And in Canada, even in North America, we have 2 important pathogens from poplar, which are Mycosphaerella popularum and Mycosphaerella poplar, which both cause a leaf spot symptom on poplar, but one of them, the Mycosphaerella popularum, is also able to cause a canker disease, whereas the other one, Mycosphaerella populicola, is unable and only cause a leaf spot symptom. So the canker disease caused by popularum led to large-scale mortality, specifically in a poplar plantation of hybrid clone, of susceptible hybrid clones, and is present, is restricted to the east and central part of North America, whereas the other species is more widespread and found all across North America. Recently, so until now, Mycosphaerella popularum, the canker disease, was absent in Western Canada, and recently we've got first report of canker on poplars in BC, in a commercial plantation close to Harrison Mills in the Fraser Valley. And so the origin of this canker disease is still unknown, but we can easily believe that it's coming from east part of North America. And the BC Ministry of Forest is highly concerned with this new disease in BC, and is wondering several questions about the widespread of this disease now in BC, and if this disease is present in natural stands of black cottonwood, which is a native species in BC, which is expected to be highly susceptible to septorial canker. So, other question from the BC Ministry of Forest concern also the possible eradication of this disease, and the source is this disease was introduced in BC through material exchanges between, for example, nursery or between research center. So all these questions were raised by the guys of the BC Ministry of Resource, and they asked us to give them a solution for that. So we use our panel of, to answer this question, we use our panel of sequenced genome, among which you have the genome of Mycosphaerella popularum and Poplicola, and we use our bioinformatic pipeline to provide some genes unique to both species, Mycosphaerella popularum and Poplicola, and finally design some assays to do some diagnostic, to do some high-throughput diagnostic. And so what is innovative with this genome-based assay is that classically for doing diagnostic in fungi, people was using one gene strongly repeated in the genome, the internal transcript spacer, but the concern is that it's not a concern, it's highly sensitive gene because it's repeated in the genome, but the concern is that to discriminate two close species, you have to work with only few mutations on this gene. And this led to some gray zone, what we term a gray zone of false positive. And what is innovative with our project is that we use some species-specific genes, some genes found only in Mycosphaerella popularum or Mycosphaerella poplicola, and we get a great amount of specificity by using species-specific genes. And to get a sensitivity almost similar to what we had with traditional approaches, we amplify and we target many and many genes. So I go back to the study, the question asked by the BC Ministry Forest Resources, and so we drive a large-scale sampling for LeSpot with the assay we developed. And for example, in 2012, 2012, we sampled almost 1,000 leaves, and we get 3.1% of positive result for the canker disease, even if the canker symptoms is generally absent in the site tested or limited to only commercial plantation. If we go more deeply in these results, we just saw that mainly this canker disease, the Mycosphaera popularum, is found only in hybrid plantation. Those found in natural population are always in natural population close to plantation. So our answer to the BC Ministry of Forest Resources was that the canker disease in BC is really confined, is in a small site close to commercial plantation or in commercial plantation, and in such case, our recommendation was that an eradication of the disease can be envisaged. So, just to conclude about all this stuff and all this project is that we try to link genomics and application to answer the different requests from different end users. And so in the example I provide you with the canker disease, we provide solution to go in a phytosanitary management to provide some advice to the BC Ministry of Resources through the elaboration of a survey and the monitoring of a specific disease. But with what we are doing, we're going to also provide some tools to help, for example, a nursery for phytosanitary certification. It's going to be specifically the case for the sudden death, for example, for which nursery going to need some certification that they exchange some healthy materials with colleagues or with customers. And the fourth task is also to provide with the genome sequencing, for example, the population genomic approach, some pest risk assessment, to be able to get some information about the route of introduction of the pathogen, information which are really important for the federal agencies, for example, to avoid to get some new introduction of exotic pathogens. I think, yeah. And just to finish, here are some examples of the collaboration and end user for our project that which are different levels. As for example municipalities with the city of Victoria about the sudden death, or federals or provincial as the Quebec Ministry of Natural Resources. All this group can be end user of our project and are asking some specific question and some specific tools to help them in diagnostics. I think that's it.